there is a chat that's on the YouTube, but you're not going to get any answers there, and you're not going to get, basically it's going to just be a bit glitchy because it's running a little bit like 30 seconds to 45 seconds behind. Okay, so I'm just going to make sure that it's muted. Everything looks like it's working. Can probably close that window, I'm assuming. Okay, so
guys, if you want to ask general questions, then don't put it in the chat, put it in the Q&A, and I'm going to sit and answer the Q&A.
Okay, so you've got general questions about,
so how long is it, and stuff about physics, and you know, what's on offer and where to find stuff. Just put it in the Q&A, and I'm going to sit and answer that, and then you can put biology stuff
in the chat.
Okay.
Okay, so I'll share my screen and then we can get up and running. Dum, dum, dum. Boom. Okay, so recording is on. I'm just going to double check because I'm paranoid. I forgot my first one. It says stop recording, which means it must be currently recording. Okay, sweet. Recording is on. Yeah, I'm not going to give you any handouts in this web class, so there will be a recording. It will be posted in the same place you used to click the link. It will be there by about 24 hours away from now. Ronnie's just told you to put general questions about other stuff, exam boards, all that kind of stuff. It's going to be all exam boards today. That can go in the Q&A. The chat can be all biology stuff. I'm going to be asking you loads of questions. I'm expecting you guys to be giving me loads of answers. And don't forget, there is a prize. Whoever's the most engaged with like quality questions, like fully attentive, asking good questions, there's someone today is going to win. The winners will be announced on Sunday at 6 p.m. I'm not going to tell you who the winner is today. I'm going to download the chat. I'm going to have a filter through it because it's too fast for me to like totally follow through all this kind of stuff. If anyone's talking on the chat that isn't biology related, we're just going to kick you out because we're not GCSE students anymore. You're here and we don't want to be wasting anyone's time, basically. Okay, so yeah, let's go. Okay, so in fact, I don't know, you've probably just seen Rich in his chemistry web class. So far, some like 6 people, I think, since Rich's web class has filled in left a review on the Facebook page. So that gives you currently a 1 in 7 chance of winning a course of your choice, absolutely free. All you have to do is leave a public Facebook review, and yeah, it's got write 1 sentence. So how long is it going to take to write 1 sentence? Not very long. So you can see basically, I think there's been 6. So if you do it, you'll be the 7th, and you'll have a 1 in 7 chance of winning it. You've left a review. Awesome, Maria. Thank you. Make sure it's public. I know loads of people have left reviews this week, actually, and they haven't made it public. So you have to click the little globe icon, I think it is, and say who's going to see this. Because if you leave it to friends and family, if I can't see it, I can't pick you as a winner. So leave a review. You are chosen at random. You're not chosen on the quality of your review. It is just leaving one. Also, you'll see here the past paper training week, tag a friend in there. Basically every friend you tag, you enter the hat to win. So we're going to pick one person and their friend, and you're both going to win the course for free. So you also have to follow the page whilst you're at it. There's loads of stuff on there that's useful. There's some chemistry, some cations. There's some biology stuff in there. There's loads of general study tips that are going to help you with your revision over the next couple of weeks. What else is there? People have been asking loads about the in fact, let's do them in order. The unlimited access, all access pass. Basically, you can invite your friends and join the course for absolutely unlimited all weekend. So biology, chemistry, maths, all the videos. There's in the course that you're all in right now. Instead of joining to click one of the web classes, you can just click to this lesson here, how to get a free all access week pass this weekend. We've exceeded our 1,000 already. So what we've done, instead of giving you access all night, we decided to just reduce the time a little bit. So it's 9 a.m. to 6 p.m. on Saturday, 9 a.m. to 6 p.m. on Sunday, but we're giving out another 1,000. So invite some more friends, and yeah, there are 1,000 more to be given away. And then basically people have been asking about where they can view some of the videos before the all access pass starts. So basically, if you go to the free resources section of the website, you can do view full course comments. So we're doing a biology web class right now, so we can click on that, and then you can choose your exam board. You've got AQA, ASNA2, OCR, ASNA2, EdXL, ASNA2. So if you buy the revision kit, this is what you get: the practical skills, the maths and the stats, the exam unlimited ultimate guide exam revision course. Basically, you get all the past papers. Basically, all that's included with the revision kit. So I'm just going to choose one of these. It doesn't really matter which one it is. You can then view, I'm actually logged in, so I can see everything. But if you're logged out, I can show you that in an incognito tab. So if you don't have the courses already, like I do, because I made them, then you can click in any one of these and you can see the full list of videos which are in there, and there are loads of them available to preview. So you can take a look at your selection. These all play. They're like real videos and all that kind of stuff. So that's how you can basically view them and get the free trial. There's no Edexcel B. There's a huge amount of overlap with the Edexcel A spec, but there's not a course specifically for Edexcel B. There's like 1,500 students in the entire country that do it, and it would take me probably weeks or months to produce it. So sadly, it's not worth my time. Okay, enough of that. We're going to come on to Amy Cuddy at the end. Without further ado, I'm going to just move Ronnie's little floating head somewhere over there so I can see. Where do I want my chat? I want my chat over here. Okay. Are you ready? You excited? Yeah. Okay, so this is a question to you first up. Command words. What do I mean by command word? And first of all, what is a command word? Explain. Okay, it's the word that tells you how you have to answer the question. This is super duper. I didn't actually ask you. If you listen to my question was, what is a command word? Not an example, and maybe can be slightly misconstrued. But okay, so we've got loads of examples here. The most common ones for sure are going to be describe. If I could write, let's do it a bit more neatly than that, shall we? And explain are going to be the two most common. For sure. Okay, what do you have to do for a explain question that you don't do for a describe question? What does explain really mean, in other words? I'm going to put this in red because it's super. Some of you guys have been here many times before. Straight to it. So give reasons for. In fact, I'm going to stop handwriting because it's not the easiest to see. So with a describe question then, you don't give me reasons. It's not looking for an explanation. And students, you're not going to get marked down for giving reasons here, but what will you lose if you start giving reasons for a describe question? Exactly, you're going to lose time. Don't give reasons. So if we look at this graph, those girls who I was in their school earlier today will have seen this basically already. You're going to be spot on with your answers. But if we have to describe this graph for 2 marks, I can tell you what the mark scheme will be for that. I remember it from a question. But describing this, we're going to get 1 for saying it increases. In fact, the mark scheme, it was 1 mark for saying it increases, then decreases. The second mark was for saying it peaks at this point here. And that was the 2 marks. There's 0 explanation needed. You really don't need it. It's just that it goes up. A describe the graph question is like, what does the graph look like to somebody who can't see the graph? No explanation needed. I'm actually going to, in fact, I'm going to skip ahead and go to my graph analysis section right now. Let's actually turn my preview off. Undo that. If you have to describe a graph that does something like this, I'm going to turn Smart Draw off. Let's say it goes. Okay, how do we describe this graph? Well, you're not going to get one this complicated. But essentially, you're going to make it, divide it into straight lines. This, we could say it's fluctuating because it's not exactly a straight line, but we're going to divide it into straight lines. So this would more than likely be a 4-mark question. We get 1, 2, 3, 4. So it doesn't really matter what the graph's doing, but you're going to just take each general point and divide it into sections, and then you're going to break it down and say, okay, it's doing this to this, this to this, this to this, this to that. That's how you describe a graph in A-level biology. And then you're going to quote points of interest. So maybe when it reaches 0, or its maximum, or its minimum, or when it changes gradient. Okay, so then if we have to explain this graph, the top graph, it's a totally different question. Now, it's giving reasons for why does the graph go up? And why does the graph go down? And why does it peak here? Why does it start at 0 here? Why does it get to 0 here? So these are all things that we can talk about. I'm not going to go into these. This is an enzyme graph. It's obviously, there's more kinetic energy, so more collisions, and that have the required activation energy. up to the optimum temperature, beyond which the internal bonds are disrupted and the active site is no complementary to the substrate, so no more enzyme substrate complexes are formed. And it's denatured. Yeah, again, squeezing all those key terms. We're going to come on to that in a minute. So I'm going to basically give me some other key terms. Not key terms, command words. Suggest is a horrible one. Suggest is a horrible one. Okay, with suggest questions, you're basically going to have to think about what it's similar to, because this is not expecting you to have wild and crazy knowledge. It's expecting you to have like solid understanding of A-level biology, but it doesn't require outside the spec knowledge. This isn't needing you to have some, it's not expecting you to know things that are outside the spec. So I'd say stop and think, what is it similar to? In fact, there was an OCR question I was looking at with the school yesterday. Oh God, I can't even remember the names of the schools I've been to. I try and remember all the students' names, but remembering the school names is sometimes I just don't pay any attention to it. And you remember what you focus on. And the name's John Fisher, I think was the name of the school. Anyone here from John Fisher? You can give me a shout out. Okay, so think, what is it similar to? Okay, and we looked at one, it was in, no, it was in Surrey, near Isha. There was a question on chitin. And chitin's in the WJC spec, but it's not in AQA, it's not in OCR, it's not in the EdXL spec. But it shows you the molecule, and it tells you that it's going to form a polysaccharide, and it's got, basically, it looks like this. Something here, there's something here, there's an OH here, there's an, in fact, I'll pull the question in, I think that exam paper's in here. And it's like, what is it similar to? It's like, well, okay, this bit is different, but what is it similar to? This is really similar to, yeah, glucose, 1 mark, it was 2-mark question, beta glucose. And so this question is not expecting you to know about chitin, but it's expecting you to be able to recognize that this is really similar to beta glucose. And when it forms a polymer, it's going to do exactly the same with as beta glucose will to make cellulose. So it is, suggest questions are, you're basically going to have to think what it's similar to, or you're going to have to make a logical suggestion. And don't get too hung up on these. We'll maybe look at some examples later on. I mean, suggest questions tend to be horrible. Like or dislike for suggest questions? Hate. Yeah, okay. Generally speaking. So a good method we can do with this is open up all the new spec papers that you can get your hands on. Go to the past paper library once you've got the course and download all the past papers in one place, because that's super handy on its own. And you can go through it and just Command F, search for suggest, and do all the suggest questions in one action hour, in more action hours in the weekend coming up. Okay, so in fact, there's too many to go through. I'm just going to mention the highlights. What do you do for evaluate questions? Whoa. This is the stuff from Ofqual, this right here. This is a full list from the exam boards that they dictate. And this is nonsense, most of this, to be honest. Their judge from available evidence is not what you have to do with an A-level biology question. In fact, I'm just going to cross that out. Evaluate questions. Screw the exam boards. I'm going to do my own one. I'm going to just pinch the one that I did from the other week. Okay, so evaluate questions. You have to give advantages or for versus disadvantages / against. So you could give, usually it's for analyzing a scientific experiment. So you can give me all the examples as to why the experiment was great. They used a large sample size, they calculated a mean, they did a statistical test. What else might they have done? But you could give me 5 reasons why the test was great. But if you don't give me one reason as to why it wasn't a good experiment, you don't pick one hole in it, you'll get a maximum 3 out of 4. They're nearly always 4-mark questions. And so, yeah, and it maybe they only tested mice, or they only tested sick patients. They didn't test it on healthy patients. So, evaluate, you've got to give both sides of the argument. Suggest, what is it similar to? We talked about that. Contrast or compare and contrast. We're looking for the differences here. If it's a compare question, you can talk about similarities. But if it's compare and contrast or just contrast, what does contrast means? Yeah, it's got to be the differences. So A versus B. And you're going to be using these comparative statements: higher, bigger, heavier, shorter, greater. You've got to have these ER words. Otherwise, it's not a comparison between the two. A is big. What does that mean? It's not a comparison. A is bigger than B. That's a comparison. Now, outline is describe the most important points. So typically you're going to get a massive section, maybe like outline the stages of respiration. Now that's an A2 topic. Let's pick an AS topic. It might be outline the stages of protein synthesis. Okay, well, they're not wanting you to go into like all the itty bitty detail of inside out of transcription and translation, because it's an outline. You just need to give the headline facts, like the most important pieces. You don't have to go into everything. Bullet pointing is helpful. We're coming on to bullet pointing. Okay, understanding them is super key, but I don't want to dwell on them too much. That's kind of relatively basic exam technique. I need to make this a bit smaller so I can see. So my questions to you. Before we start writing our answer, we've got the question in front of us, an imaginary A-level biology question. What do we do before writing the answer? Okay, you're going to highlight the keywords, understand it. Teachers often say, read the question twice. I mean, good advice. You should read it more than once, but like read it carefully, definitely. I'm going to give you some specific actionable things that you actually need to think about in order to get this kind of in your head. So the things that we need to know before really cracking on and starting our answer is, what is the command word? Which tells us, how are we going to answer this? Is this a describe, this is an explain, is it an evaluate, is it suggest, is it compare and contrast? That's super important to know. What else have we got to know? Marks, I'm seeing, absolutely. Now, I'm going to show you some examples of how I answer some questions later on, and I haven't really looked at the answers, so we'll see how it goes. But marks, this is what tells me how much I need to write. The number of lines or space available, you might get all of this for 4 marks. Well, 4 marks, I can answer, as long as it's not a quality of written communication question, which some of you OCR students have, 4 lines, maybe 5. I might cover my arse by putting an extra line or 2 in. But like effectively, all of this is not a guide for how much you should be writing. A lot of students just start writing and they just keep writing until they get to the end, because they think if they fill the space, they're going to get more chance of getting the mark. That is nonsense. It's just, you need to be writing your answer to the level of detail that the marks give you, but not to the amount of space available. Sometimes there's an obscene amount of space for a question. Okay, what is it actually asking? That is definitely something we're going to work on today. I'm going to call that the topic of the question, because there's a reason I need to know the topic before thinking about the answer too much. Because once I've got the topic of the question, and this is tricky, so be a bit careful with this. But we're going to do some work on that in a minute. We're going to come up to this. Now, once we know the topic, let's say, give me a topic, an AS biology topic, because we've got some AS students in here. I don't want to alienate people. Enzymes. Okay, enzymes. Let's go. Enzyme specificity is maybe one that we can choose. A classic, I can tell you a question off the top of my head for enzyme specificity. I'll give you the question and I'll tell you how they gave it to you in the exam. I'm going to just change color. So the question was, maltose is hydrolyzed by the enzyme maltase. Okay, statement. Explain. What does explain mean? Explain why maltase catalyzes only this reaction. Okay, so this is basically how can we decode this sentence? What does this question actually asking us to do? If we look at this, the topic of the question is enzyme specificity. Let's maybe put these in the margins. The command word, which is explain, which is equal to give reasons. It was 3 marks. I can tell you that off the top of my head. topic of the question I'm going to say is enzyme specificity. Why are enzymes specific? Okay, so we're then going to basically get the question super clear in our head. So we're going to, without being able to read it, I need to know what this question is. And this is really important for a couple of reasons. So because writing is what we call it in language teaching, a productive skill. A productive skill is something that we produce ourselves. So it's like when you were speaking, maybe we learned French GCSE. It's easier to understand French because there'll be words that are similar to English words. That's a receptive skill. I'm taking information in and I'm decoding it. Writing and speaking are productive skills. So the information has to start in our head and come out of our pen. And therefore, the question has got to be crystal clear here. I read answers all the time, and it's obvious the student hasn't thought about it. Not that they didn't know the answer, but they haven't got the question clear, because this is the column of where they should be writing, and they start and their answer just drifts off and off and off and goes way off topic. So I'm going to say get the question clear in your head, and then we can rephrase the question so it makes more sense to us. Get the question clear in your mind. Oh, it's difficult to write. I'm going to buy an iPad, Ronnie. I'm over this graphics tablet. It's not doing it for me. Okay, what else can we say? Get the question clear in your mind. I'm going to then say reword the question to yourself, which is how you're going to get it clear in your head, basically. To make it easier. This takes a bit of practice, a little bit of skill. We're going to show you how to do it. And then number 6, what we're going to do is brainstorm. Again, we haven't started writing yet. All the relevant key terms. Okay, so all the relevant key terms. Okay, so how would I do this to this question here? It's a bit of a sloppy example. Maltose is hydrolyzed by the enzyme maltase. Explain why maltase catalyzes only this reaction. So what are we looking? We're looking for give reasons why maltase, an enzyme, catalyzes only this reaction. So explain is give reasons, 3 marks. The topic is enzyme specificity. So again, I don't have to close my eyes here because I can just do this. Just make it all go away. Okay, so now we can get is the question clear in your head? Is the question clear in your head? Okay, so we can reword it. So if I was rewording this to make it as easy to answer as possible, obviously you still need to be able to get full marks on it. I would say, give 3 reasons why enzymes are specific, and then maybe using maltase as an example would be how I would phrase this. If I wanted you to get full marks, give me 3 reasons why enzymes are specific to one substrate, and we can use, if we're going to use an example, we're going to use maltose and maltase. Is that easier to answer, or is that harder to answer? Okay, so it takes a bit of skill and takes a bit of practice to be able to understand the questions and demystify them in this way, but literally past papers are the key to this. I've done, no joke, over 1,000 past papers, A-level biology past papers. Like easily 100 a year for 10 years. And that's how I know the language of A-level biology papers, is because it's experience and it's practice. But you guys can get way better in the next month. It's a month tomorrow, you are finished paper 3. This time in a month tomorrow, paper 3 is finished. Okay, so brainstorm all the relevant key terms. I say, what are the relevant key terms here? We've got enzyme, tertiary structure, maybe hydrogen bonds, disulfide bonds. We've got primary structure, maybe. We've got maltose, we've got maltase. This can be loads, yeah? But I'm just going to brainstorm all the relevant key terms first. Then I'm going to pick the most important ones. So we've got active site, enzyme substrate complex. Yeah, all these things. You can brainstorm all the relevant key terms, and then we're going to pick the most important ones, and we're going to put them in a logical order, and then we're going to write Now we're going to start writing. Only at bullet point number 7 here are we actually going to start writing. I'm just going to move down a fraction. Okay, we're going to write bullet points. Unless it's the essay question for AQA, unless it's a quality of written communication where you get marked by level for the other exam boards, you can write in bullet points. Now, this is super important. Writing bullet points, I'm going to say 7 to 10 words. Short bullet points. Oh, again, girls from St. Philomena's will know the answer to this. I did an analysis of it with that. I did OCR, EdXL, and the AQA separately. How many words are there per mark on the mark schemes, roughly? 4.5, 5? You girls are straight in there. Okay, so there's about 4 point something words per mark on the mark scheme. So we don't need to be writing a lot. And if you shouldn't be trying to capture more than one mark scheme point in a sentence. Each sentence needs to be designed to catch one mark scheme point. Each sentence needs to answer the question in some way. Otherwise, it's not going off topic. So in bullet points, 7 to 10 words. At the end of your full stop, you are a new, clean, and what needs to be in each bullet point? Exactly, one key term. So if you're finding you've got lots of commas and semicolons and ifs and buts and ands and maybe, and you've got like a sentence that's got 3 or 4 different key terms in it, try and practice this to your past papers. Train yourself to be like one sentence, one bullet point, one key term, full stop, next point. And that the reason why you need to think in bullet points is because you need to complete that sentence in your head between. You don't just carry on writing at a full stop. You don't just like keep on sprawling out words. It's one sentence. You think about it beforehand. You get to the end of the sentence, full stop. And then what's the next bullet point? You shouldn't be writing continuous flowing prose because that's just a recipe to go off topic. And you want to be going like point 1, point 2, point 3, point 4, check out, next question. Yeah, you do get about, the exam boards vary depending, and the exams will vary. You get about 1.2 minutes per mark. So almost, 1.2 minutes. So it's about 70 to 90 seconds. So up to 1.5 minutes per mark. Okay, so we're going to look at applying this. Okay, so here's the question. Reactions involved in photosynthesis are affected by environmental factors. The graph shows the effect of temperature on the rate of photosynthesis in wheat. Explain the effect of temperature on the rate of photosynthesis in wheat. This is an EdXL question suitable to all of you guys. What's the topic of the question? 1, 2, 3. We've got command word is explain. 3 marks. It takes no time at all. Some photosynthesis, some enzymes. Okay, so next up, we've got the topic of the question. We don't necessarily know at this point, limiting factors. Well, the graph is a bit of a clue here. And actually in the exam, these were on a double page spread. So it wasn't exactly next to each other. I've made it slightly easier by putting the graph right next to the question. Okay, if you had to bet one topic, is this photosynthesis or is this enzymes? Enzymes. This, despite the fact we've got all this nonsense about photosynthesis going on here, this requires us to know that photosynthesis is an enzyme catalyzed reaction, or in fact, many enzyme catalyzed reactions. But this could be respiration, this could be transcription, this could be DNA synthesis, this could be any reaction that goes on inside a living organism, basically, because more or less everything is catalyzed by enzymes, more or less. We might want to mention Rubisco, maybe, but it's probably not a required, because what's the question asking us? Look at the question. Explain or give reasons for why temperature affects the rate of photosynthesis. And so why does temperature affect photosynthesis? If we get down to it, it's because of increased kinetic energy. It's an enzymes question. It's got basically nothing to do. We don't need to mention Rubisco. Would be maybe a nice idea. We could throw a Rubisco in for sure. Okay, now what are the relevant key terms then? same as, basically, it's the same as the one I just made up off the top of my head before. So we've got enzyme substrate complex, we've got tertiary structure, we've got substrate, kinetic energy, denaturing, optimum temperature. You guys are smashing it. Be careful with collisions. They want you to say the number of successful collisions or collisions with the required activation energy, but that's a bit of a mouthful. So successful collisions is fine. It's true that there are more collisions, but in terms of the relevance to this question, there's more collisions with the required activation energy or successful collisions. Super nice. Okay, we're moving on. We're getting smashing through this tonight. Okay, so this is some practice on rewording the question. Do you talk about induced fit in lock and key? For that question, not relevant. It's because the question was, why does the rate increase with temperature? So that's not relevant. Lock and key or induced fit, that's a model for how enzymes work. The question was, explain how the enzymes in photosynthesis are able to catalyze the reaction. The question was, how does temperature affect the rate of reaction? You definitely want to be top three key terms for that question would have been active site, enzyme substrate complex, and then kinetic energy or select kinetic energy linking that to successful collisions. Okay, let's look at this one first. Let's just do one at a time. Let's go to blue. This fat is a substitute. This fat substitute is a lipid. Despite being a lipid, it cannot cross the cell surface membranes of cells lining the gut. Again, it's a suggest question. I don't want you to answer it. I want you to reword the question. Suggest why it cannot cross the cell surface membrane. If we want to strip away all the nonsense about this and make it as easy as possible for someone else to answer, how can we transfer the, it's only 1 mark. The question I'm thinking of would be more than 1 mark, but what is it similar to? Suggest why it cannot cross a cell surface membrane. Suggest why lipids are insoluble. Why can't it leave the cell? So my rewording this question would be like, I would say, why can't some molecules diffuse, or I can't write, through the cell membrane by simple diffusion? So this, we don't know for sure, but we're not expected to know what this fat substitute is. We don't necessarily know how big it is necessarily, but we can give any sensible, biologically sound reason why a molecule isn't capable of traveling across a membrane. The only thing we've got to be careful of here is saying that we know that it's a lipid, so it is fat soluble, which will be, but it could have a polar charge to it. So there's why can't something cross the membrane? It's either too big, it's either polar. Anything else? Basically, they're the two main ones, right? It will need a specific protein. Polar means it has a charge, has poles. There's a positive end and a negative end. Yeah, so like the poles of the planet, that's what polar means. Its shape isn't complementary to a protein, a transport protein, or transmembrane transport protein. Yeah, so basically, this question is not that difficult, but it could be confusing because we know that lipids do diffuse through the cell membrane typically, but only small ones. So it could be too big, it could be polar, or maybe there's no transport membrane. Okay, let's look at another one. I've had to say this is like part B of the question. Part 1 told us about the tissues. These tissues in this question is xylem and phloem, just so that we know. Okay, again, I don't want you to answer it. I want you to reword the question so that we can make it easier to answer. And again, we can change it quite a lot. The roots of this sunflower plant were put into a solution containing a metabolic poison. Whenever you see metabolic poison, it means a metabolic poison prevents the production of ATP. So this is our clue. Explain, and again, we can remove explain from it and replace explain with give reasons for. Explain, give reasons for how this would affect the transport of substances in these tissues, by which we mean xylem and phloem. Okay, so the roots of this sunflower plant were put into a solution containing metabolic poison. Metabolic poison prevents the production of ATP. Explain how this would affect the transport of substances in the xylem and the phloem. Okay, give reasons why transport needs ATP. Yes, there's for 3 marks, I could be maybe a little bit tempted. Oh, that's not what I wanted to do. I think we need to reference both of these tissues. Specifically mentions both of these. I would, in my head, I would make this a 4-mark question and do 2 on the xylem and 2 marks on the phloem. If I can. It might not be completely equally weighted, but that's what I'm doing. If I've got a question that kind of splits down the middle and it's a 3-marker, I'm in my head going to make it, okay, I've got 4 things to get here. I want to try and do 2 for this and 2 for that. Okay, so how would I say this? I would say, give reasons for how transport will be affected in the xylem and the phloem if no ATP is available. Okay, and then we can now that we've understood that, answering it becomes way more straightforward. Xylem will be unaffected because it's made of dead tissue. It's passive. It's probably for effectively 2 marks. That's all we need to say. Xylem will be unaffected. It's dead tissue. No ATP is required. There's no cytoplasm involved. Again, crowbar in all those key terms. Dead, no cytoplasm, no ATP required. Okay. Then we can say phloem will be affected. Why will phloem be affected? Because it requires active transport to pump the sucrose or the assimilates into the sieve tube cell. And without ATP, there'll be no active transport, which means there'll be no change in water potential, which means there'll be no movement inside the phloem. That make more sense? So spending a little bit of time, especially in your past papers, spending your time analyzing these questions and thinking, okay, I can see all the fluff here. Now, this is easier for me because basically what I do is like I answer the question in my head and then I reword the question to match the answer, which is effectively the skill that you're trying to do, but it does take a bit of practice. And obviously you've got to know it kind of well too. But that doesn't mean that you're not going to make massive gains by doing this with every single past paper question. I mean, if it's a 1 mark, if it's like, name this bond, then it's a peptide bond. You don't need to analyze the question and, give, state, name, and calculate are not command words which are going to trip you up. You might not be able to do the calculation and there's no excuse for that, is there, Ronnie? Because calculate questions, you can practice calculate because maths is easy. All you've got to do is like practice the questions because the question's always the same. Calculate chi-squared. Yeah.
That's easy. That's why people with maths A-level earn 10% more than people without maths A-level because it's just so easy.
Yeah, definitely. But with the calculate questions, you can just open if you're struggling with those calculate questions, open up all the new spec papers from all the exam boards, AQA, OCR, EdXL, all the stats is the same, all the maths is the same. Search for the command word calculate and just do all the percentage change, all the volume of the spheres, all the surface area of the circles, do all of them in 1 day and mark them and check them and go back with the technique I'm going to show you at the end of this web class and you're going to get way better. You're going to get way better. So if you know you've got a weakness, that's what you need to be working on right now. That's what you'll be working on. Don't test yourself on the stuff that you know because it makes you feel good, which means, oh, yeah, I know all this. I know these biological molecules. They're wonderful. But if you know you struggle with the maths bit, they're the things you've got to push yourself in the next month. You've got to really challenge yourself to do the things that you're not so good at. Okay. Let's have a crack at rephrasing this question. NMO is a disease that leads to damage of nerve cells in the spinal cord. Probably completely irrelevant, to be honest with you. Spinal cord may be worth noting. A person with NMO or the disease produces anti-AQP4 antibody that attacks only the nerve cells. Explain why all the anti-APQ4 antibodies only damage these cells. 4 marks, give reasons for. Okay, what's the topic of the question? Topic of the question. Specificity, protein specificity, but can we say enzyme specificity? It's not an enzyme. Does that bother us? Does it change our answer? Apart from the fact we can't say active site and enzyme substrate complex anymore, the answer is the same. The answer is the same as it was for the maltose and maltase question. Give reasons for why the antibody, the protein, is specific. Only damage. It's the same as the maltase question, exactly the same. And I bet you the marking points will be identical, apart from the enzyme being specific. It's going to be the antibody is specific to the antigen. Yeah, where's that antigen found? It's only on these types of nerve cells. But the answer is exactly the same. So we can say, give reasons why an antibody is specific, using these words as examples. But these words are basically completely irrelevant. We don't need to know any of that. All we need to know is why proteins, well, they've got a specific tertiary structure. Yeah, antibodies, they've got the variable region, yeah, which is complementary to the antigen, and therefore they form an antigen-antibody complex. You got it, guys. You got it. Okay, so let's zoom out just a fraction. Okay, let's look at the mark schemes just in case you're wondering. Suggest why this random lipid cannot cross the membrane. We've got hydrophilic, is polar, is too large, is too big. So this question is, why might a molecule not be able to diffuse through the phospholipid bilayer? Any of those reasons is valid, because it's a suggest question. What is it similar to? Something that can't move across the membrane. Okay, why can't something move across the membrane? Well, for any of these reasons. Any of them is acceptable. Ignore is not lipid soluble because it tells us it's a lipid. The root of the sunflower plant. Okay, so I've put the wrong mark scheme in there, haven't I? I wonder if there's one underneath that. You can just take my word on that because I just know these things. Okay, so xylem unaffected because it's dead tissue. There's no cytoplasm, and phloem will be affected because it can't use ATP. Therefore, it can't do active transport. Therefore, it can't pump sucrose or assimilates into the sieve tube cell, and therefore there'll be no change in osmotic pressure, water potential, and therefore there'll be no hydrostatic change. And then the one we just looked at, it's a specific tertiary structure. Tertiary structure, key term, has a binding site. It's complementary to one antigen. It's only found on these nerve cells, and it forms an antigen-antibody complex. Again, you're looking to mimic these mark schemes as much as possible. If you're writing long sentences, again, look, how much space did they give us for this? How much space did they give us for this? Like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 lines. You should be looking at trying to do one bullet point per line. Specific tertiary structure. If I'm going to show you how I kind of write my shorthand bullet point answers in my past papers. Specific tertiary structure. We can say complementary to antigen. And then in my notes, I'd be antigen antibody complex. I hope you can read my writing. Just about. This is probably how I would have bullet pointed that in my answer. And obviously in the exam, I'm not going to write quite this ridiculously shorthand, but it's important to smash through as many past papers as possible. If you can make a past paper go by a bit quicker by bullet pointing your answers, I can put this in a full sentence. This isn't a challenge to me. The protein has a specific tertiary structure, full stop, which is complementary to one type of antigen, full stop. It only forms an antigen-antibody complex with cells with this nerve, with this antigen, full stop. Okay, moving on. Next question. Oh, I think you might, whoa, how do I zoom out? Okay, so this one I'm going to challenge you to do. We're going to analyze the question first, then we're going to go 5 words per mark. Okay, so a scientist investigated the effect of treating leaves of one of the species of plant with 3 different substances. These substances reduce the stomatal diameter. He compared the mean diameter of the stomata after treating the leaves with these substances. I read that terribly. He compared the mean diameter of the stomata after treating the leaves with these substances and the mean stomatal diameter on the controlled leaves treated with distilled water. scientists' results shown in figure 6, the mean stomatal diameter of the control leaf was 7.5 micrometers. Check the scale, make sure there's nothing too strange going on. It's not continuous data, so it's a bar chart. Reduction of mean stomatal diameter compared to the control. Okay, so here, it's a reduction compared to the control. So how much is it less than 7.5, which is a bit of a weird way of representing the data. It's completely okay for them to do that, but this doesn't mean that the reduction was nearly 3. So it'd be 7.5 micrometers minus whatever the value is off the bar chart will give us the total diameter of the stomata, which maybe is a question we need to know about, maybe it's not. ABA is a substance that some plant species produce when little water is available. Explain why producing ABA may help these species survive in dry conditions. Okay, so again, we're going to analyze the question like we do every single time. 2 marks, it's an explain question. So now we want to give reasons for why. Forget producing ABA, because ABA, I don't know what ABA is. I've never heard of it until I did this exam paper. So again, it's not expecting you to know stuff like this. What does ABA do? Well, that's over here. Yeah, these substances reduce the stomatal diameter. Okay, so in my head now, I'm thinking, give reasons why a compound that reduces stomatal diameter will help species survive in dry conditions. That's not rocket science. All I'm doing is just scrubbing this bit out of the question and substituting in the bit that they've told us that ABA does. I've not done anything magical there. So we want to give reasons for why a substance that reduces stomatal diameter helps a plant survive in dry conditions. Okay, so now we've got 2 marks to get. So we're thinking about stomata. Topic of the question is sort of benefits of water conservation to a plant. Brainstorm all the key terms. Okay, so we've got maybe guard cells, flaccid, we've got water vapor, we've got why does a plant want water? Why does it not want to give away water? What is water ultimately used for? Transpiration, great key term. Evaporation, good key term. Photosynthesis, can we be a little bit, photosynthesis is probably going to get us a mark here. I would like to be a bit more specific with our key terms. So I'd say, okay, photolysis, super nice. This is an A2 topic, so any AS students don't freak out too much. This is very much an A2 question. Explain why, give reasons why something that reduces stomatal diameter help the species survive in dry conditions. So I could say, I'm going to say it reduces, 5 words per mark. Okay, I'd say reduces transpiration. Full stop. I wouldn't actually do very much different in the real exam there. I'd say it reduces transpiration maybe, which maybe conserves water, H2O, for what I'd write in my past papers. We can maybe say H2O used for photolysis. In photosynthesis. Cool. In fact, what I might say is in the light-dependent reaction of photosynthesis. Again, just find an excuse to throw that key term in there. Okay, so like it reduces transpiration, full stop. It saves water, conserves water, full stop. Water is used for photolysis. You could probably get away with a comma there, but like, again, I'm thinking of it as a single standalone bullet point. Water is used for photolysis, 1 key term. Okay, now I'm moving on to my next sentence. What else could be on the mark scheme from the light-dependent reaction of photosynthesis? And that's loads for 2 marks, but it doesn't take long to write this. Anyway, anything lysis means split? Absolutely. Photo means using light. Anyone want to add anything to that? Have I missed any key terms? Let's have a look at the mark scheme. Less evaporation, less transpiration. In fact, nearly nailed it. Less transpiration. That is it. Marking point 1. Less transpiration. Marking point 2, more water available for photosynthesis or metabolism or any other metabolic reason. Obviously, water for condensation, for a hydrolysis reaction. less water needed from the dry soil. Okay, interestingly, if we just say it saves water, no mark. Saves water, e.g., always give an example, e.g. for hydrolysis reactions. We give the example, we get the mark. If we don't give the example, we don't get the mark. So giving examples is going to help you with picking up those marks when you're not quite specific enough with your terminology. Okay, where's my other questions? Let's have a look. Oh, this is a tricky one. Okay, so I'm going to answer this in 5 words per mark. Again, you can do more than with this question here. You can do more than 3 bullet points, but I want each bullet point to be short and concise. But again, we're not going to answer it just yet. Let's look at the question. Let's analyze the question, because this is where the skill is. When HIV infects a human cell, the following events occur. A single-stranded length of HIV DNA is made. Okay, I know what's going on here. This is reverse transcriptase, taking RNA and making it into DNA. The human cell then makes a complementary strand of the HIV DNA. Okay, so this is it's in DNA form already. So it's going from single-stranded DNA, which is a bit weird, into now double-stranded DNA because it's the other half. The complementary strand is made in the same way as new complementary strand is made during semi-conservative DNA replication in human DNA. Describe how the complementary strand of HIV DNA is made. Okay, so what do we do? First up, 3 marks, describe as our command word. Okay, this is not straightforward. This is elements of DNA replication, and we could just start regurgitating DNA replication. DNA helicase separates the hydrogen, separates the strands, but would that be right here? Would it be right to say DNA helicase separates the hydrogen strands? No. Why not? It's already single-stranded. It's a weird one. Okay, so let me draw this because I looked at this question with some students a while back and they got really confused here. So we've got our HIV, its weird alien legs attaching onto the cell surface membrane. Okay, and I'm going to do the RNA in my notes, in my course. RNA I do in red, so it's got the RNA. So the RNA is injected into the cell surface. So this is point 1. A single strand length of HIV DNA is made. Okay, so now this is the RNA. The enzyme reverse transcriptase is going to read this along, zip, 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 and we're going to make some HIV single-stranded DNA. So now this one is DNA. It's actually complementary DNA because it's made by reverse transcriptase. This is basically our messenger RNA that's coming from here. So we've got this single strand. And this question is asking us, if we think about the question, this is saying the human cell then makes a complementary strand of this DNA. So this is made into a complementary strand. So it was starting with this piece here and we're making it into this piece here, which is why we can't say the DNA strands are separated by DNA helicase. It just shows that you haven't taken the time to break down the question to get it there. Okay, so how is this done? It's the same method, yeah? So what are we gonna say? We're gonna say, let's do this, I'll do it in blue. We can say, what do we need to mention? Enzymes, DNA, polymerase, adds bases. I would probably say nitrogenous bases, but I'm trying to do it in fewest words possible. Yeah, by complementary base pairing or specific base pairing. Give an example. A with T and C with G. What else we need? So the enzyme forms phosphodiester bonds. Probably got enough there for 3 marks, but What else might I want to say? Yeah, one single strand acts as the template. Yeah, we can't say both strands act as a template. We don't have both strands. So we can say the single DNA strand equals the template. In my past papers, you're allowed to do stuff like this. Again, what we're trying to do is get that nugget of information onto the page. So I use lots of shorthand and stuff like that. I know I can put this in a full sentence in the real exam. I will definitely, don't let me make you think that this is how I would write in the real exam. You need to put this in a sensible sentence that's understandable in the exam. But in the past papers, we just want to be fast as we can. I do lots of like increase in temp equals increase in rate, more enzyme substrate complexes. So that's how I'm generally writing in my notes when I'm like doing like past paper questions, because I want to do that past paper as fast as possible. So I want to mark it and find out what I've done wrong so I can learn all the bits that I've done wrong so I can remember them for next time. Everyone happy with this question? Bueno. Okay, what else? Okay, what have I done there? What have I done there? I've zoomed all the way out. No, I can't. I'm going to use my mouse for once. Oh, it's just slow, just being slow. Okay. What do you need to know about the exam? Okay, so dates. Put them in your diary. You need to know when these things are. If you're doing the AS exams, the 24th and the 4th. Not anyone here doing the AS exam? There's 300 of you in here. A couple of you. Okay, so they're your dates. Make sure you know when they are. You know what's on each one. And then most of you are going to be A2 students, I'm assuming. The 7th of June, the 11th of June, and the 18th of June. Yeah, so it's the 18th tomorrow, a month tomorrow. You're done with A-level biology. Am I doing an A2 webinar? Yep, tomorrow night. I'm going to be in London after catching up with my Ultimate Guide subscribers. Yeah, it freaks you out. Well, you've got a month. You can still learn a lot in the month. Like we've had students complete the entire A-level biology course, my course, in a week. I mean, you can easily smash. I mean, I say easily. You can get through the entire course in a week. I'm doing a whole load of web classes on paper 3 and stuff like that. The essay, if you're an AQA student, all that kind of stuff. Okay. Who uses the spec like as religiously? Who uses the specification as like it's your Bible? Okay, so who doesn't? Nope. Okay, so like the spec, it will tell you in theory everything that's going to be on the exam or the topics. They're less useful than they used to be. Specs used to be awesome. It used to be like you have to know all of these little bits and it would tell you specifically what you need to know. And if it wasn't there, you didn't need to know it. You kind of just had to learn that document and you were kind of cool. Now it's really vague. It doesn't tell you the level of detail that's absolutely essential. Why not? Because they don't want it to become a memory test. But that just makes it unfair because now they don't tell you exactly what's going to be tested. Well, they're testing the gaps between the things that they've told you. I actually got an email, I'm not allowed to show you my inbox, it contravenes privacy policy. Maybe I'll copy and paste it. Maybe I'll pause my screen sharing from the AQA, I think it was chief examiner, or a message from them, because I'd asked them a question about resting potential, because there was some conflicts between some of their exam paper, and I was like, these are not consistent. And okay, I'll pause my screen. Pause share. Ronnie, you just have to tell me, are you still seeing the whiteboard? Okay, sweet. I'll open my inbox just quickly.
Yeah, I can't see your inbox.
Okay, sweet. Yeah. I'm just going to copy and paste this. It's, maybe can take a screenshot as long as there's no email addresses in it. From Teacher Services at AQA. Specification, terminology, clarification. Okay, I'll just take a screen grab. It's going to be the quickest, I think. Elise, I'm allowed to have a name in there. Yeah, that's fine. This is, yeah. I haven't read it in full yet. I read the first like 2 sentences. Resume share. Okay, I can do this. Zoom panel's going to go away. Okay, let's drop that in here. Let's make this big again. Okay. We don't have my original email. Hi, Richard. The Chief's reply to your questions is below. I hope it helps. There are different types of ion channel. So this is, I asked them specifically, according to AQA A-level biology, about the ion channels, because there was a bit of disagreement. So there are different types of ion channel, including passive ion channels, also known as leakage channels. This has never been in a mark scheme ever. Never seen that in an A-level biology paper ever, which allow specific ions to diffuse through the members and are always open. Okay, this is not terminology that's ever been on the exam. So why are you telling me? I can look, I know what the science is. You don't need to tell me what the biology is. I know the biology. I want to know what you think the level of detail is specifically for the exam so I can teach my students that. Active ion channels are gated and can be open or closed. Interneuron passive channels for potassium, like that was the first thing they said, show me. It's like I can't believe I've asked you in the context of your exam, what do I say? If I use this on the exam, how many marks am I going to get? None. Precisely none for talking about any of this. This is nonsense. Like, yeah, anyway, you can read it there if you want to. I was not impressed. One day I'll speak and AQA will listen, or any of the exam boards will listen. Okay, so that was just a little rant. Yeah, I'm getting mad. It's just so frustrating. Like, it's hard enough for me, and I spend my life doing this. I feel so sorry for you guys in that you've got an almost impossible hashtag leakage channels. Yeah, tag AQA in that, and like if you write that in your exam, like you won't get the mark. So don't write it in your exam. But yeah, it's just crazy. Okay, so these are the dates. Have a printed copy of your spec, because the spec should tell you a little bit as to what you need to know. And because there's loads in the textbooks that are not going to be in the exam. I was working with the OCR textbook the other day. I opened it live, and it's like MHC, major histocompatibility complex. There's no questions on that. Why is it in the textbook? Or why is it not clear in the textbook that this won't be in the exam? They can't ask you about MHC because it's not in the spec. Crazy. You got to know, like, knowing the spec is super handy though, especially for AQA students, because they have to write an essay and they have to draw from different parts of the spec in order to get top grades. Less important for everyone else, but knowing what's going to be on into these exams, especially the OCR and EdXL students who their paper 1 and paper 2 is going to be a mixture of different modules, and you need to know which modules are going to be on the exam. Okay, timings. Yeah, no, these papers vary in length, and it will vary by exam board. So just open up your past papers and check, how long you've got per mark in each of those papers. A-level biology exams, I'd say this all the time, test you on how good you are at A-level biology exams. So the one thing you can practice the most to improve your performance, what should you be doing? I think there's two things that you should be doing to fine-tune your performance over the next month. You need to pass papers and use my videos. They are the two things that are going to be the fastest way of improving your exam technique.
Rich?
Yeah.
Can you talk about how to use the past papers? Because a lot of people on the chat are asking me about
Saturday
the week and what to do. And
that's in Saturday's web class. That's because we can apply that to all subjects. So yeah. I'm going to do that on Saturday.
Okay, cool.
So what you need to do is use the exam papers as much as you can, because they're going to be the thing you're actually tested on. So they're super duper important that you do that. In fact, I might even just pull this off so you can just see me. I might also just make sure I can still see my notes. Okay, so that's what you need to be doing, is using past papers. Also, the fastest way, if you've got areas that you're not too hot on, you don't have very much time left. How long does it take to read a chapter of the textbook and write notes on it? What would you say? Ages. Like a full chapter, let's say you don't like the immune system. You want to go back and just redo all of the immune system. Aeons, 2 hours, 4 hours, 5 hours, half a day. Yeah. Snap, revise, or tailor tutors? Ask the room. You guys can answer that question. I'm not going to tell you. Snap, revise, or tailor tutors? Yeah, well, yeah. Yeah, we'll check out the videos. But okay, how long would it take to watch? I think the immune system is T cells, B cells. I think there's 5 videos in that module. So they're about 8 minutes each. So how long does it take to watch 5 videos and take some notes down? Probably an hour to 2, probably, if you're doing it really well with notes and stuff like that. I'd say 40 minutes and 20 minutes, something like that. Not too long. So like you're going to cut your revision time compared to a textbook by about 1/3 is roughly what I reckon. By the time you've taken notes to my videos, it's probably going to be about 1/3 the amount of time it takes you to do it from the textbook. Also, I'm going to talk you through it. You're going to get me effectively sitting there with you, guiding you through whichever topics you've got issues with. You're going to get taught it from me, and I am not too bad at this. Like the only reason I started doing this is because I used to get students. I used to basically add 3 grades to a student. If you paid me as a tutor throughout the year, I'd get you 3 grades extra from where you were predicted. And like I've basically put that in a course, and now you can buy it. Crap, revise. You're welcome to start trending that as a hashtag as well. Love it. Okay. Also, top tips for the day or the night before. I often get asked questions like, what should you be doing the night before? Like, ideally, you've got a month now, and then this month is important. So you want to be using that month with the past papers so that you don't need that last 12 hours. There have been times when I have done an all-nighter and I've needed that time to get the information in my head. That is not an ideal situation at all. And I'm not telling that because I'm your mum and dad. I'm telling that because I'm a killjoy. I'm not telling that because I'm your teacher. Like, generally speaking, pulling an all-nighter the night before is not what you want to be doing. You've now got 31 days until your exam ends. So there's plenty of time. That 12 hours is way better spent sleeping than it is crashing content in. You should be motivated enough by now. You've done 2 years of work. You've got set yourself some targets, and the web class on Saturday is going to be about how to use those past papers more effectively, how to plan your time during half term coming up, basically how to maximize your past paper use between now and the exams. Also, what you need to be doing in the run up is using those mark schemes. So you're like tightening up your answers and every time you don't get full marks on an answer, you need to be adding those answers to a list, to your notes, so you can learn from your mistakes. Basically, you need to be learning the stuff that you don't know. Really common for students just to keep testing themselves on the stuff that they know because it feels nice. Yay, I got 99% because I've just revised it, and I just keep going back over the stuff that I get good marks on. But what you need to be doing is testing yourself on the stuff that you don't know, because if you do get some of it right, it's like, oh, actually, I don't have to revise the phagocytosis bit of the immune system. All I need to do is it's the T cells and B cells, or it's the active and passive immunity, or it's the primary and secondary response, or it's the vaccines. You're going to find out what you don't know about, and then you're going to use the videos to go and smash it, basically. Okay, what else would I suggest? I did a bit of graph stuff earlier. Also, the examiner, Ronnie's an examiner, and she can tell you about this. This is basically when they've got your scanned copy, you've got maybe your writing is like this. Maybe it's a long question. You've got all like this. And basically, they're going to get a window that they can like scroll up and down. So they don't even get to see your whole page. So they get to go down, and if it's like, if it's hard to follow, they can only see this little window, and they've got to go up and down. So it's really difficult for them to keep track. If your answer is scattered all over the place, if it's a sequence, start at the beginning and lay it out in those bullet points with your key terms that are going to make it easier to follow. Yeah.
I can't see your screen.
Okay. Yeah, that's a good point. I was getting carried away with myself. Let's go share screen. Okay, so this is our page on the right-hand side, and this is this little window that the examiner. Is this more or less accurate enough, Ronnie? Like where you can just scroll up and down?
It is for me. I mean, obviously, I haven't marked biology, but I don't know if they use the same system, but I would assume they use the same system. The scripts are all scanned in, and then you mark like a billion question ones, a billion question twos, a billion question threes. You don't mark somebody's script. So you don't have like the whole paper. You just mark loads and loads and loads and loads and loads of question ones. And then at the bottom of the screen is where you put the marks. So if it's if it's like out of 3, then I can extend the amount of script I can see, because I only need a little bit of space for the 3 marks. But if it's out of 15, then I need 2/3, 3/4 of the page for marks. And then I can only see a little bit at the top.
So a couple of questions. I was just watching the chat a bit there. So with the recording again, will be up this time tomorrow. If someone said they're having issues with audio,
but you can't say things like go to page 8. Well, I mean, like you could, but your script would have to be pulled from the thing, and it would, you know,
and all the rest of it. That was a really good question there. How many hours a day? I mean, I'd say we're going to look at some of that on the revision web class on Saturday. I'd say if you've got a full day, like during half term, which is coming up, I would say then, yeah, 8 hours is probably a pretty solid work day. Has anyone been using action hours? The action hours that we suggested, we've been doing a lot of this. It's all going to be in the web class on Saturday. Okay, so I'll give you a little sneak preview. This will be in the web class on Saturday. Let's just draw myself a little circle. So this is 60 minutes. Who's sort of the Pomodoro technique? So Pomodoro is 25 minutes working, 5-minute break. There's a few things you need to do with this, which is slightly different. This is a 45 minutes working and a 15-minute break. Because 15 minutes is actually half useful. You can do something in 15 minutes. 5 minutes is kind of useless. And this is really going to help with your motivation as well, because you can set yourself at the beginning of the day, okay, I'm going to do, let's say 8. 8's a full day. I mean, yeah, 8 is loads, but you can then do, let's say you're doing, you know, you need to prioritize your biology a little bit more. So maybe you can do 4 sessions on bio. You might want to do 3 sessions on your maths. And that leaves us 1, maybe you're cruising psychology. And so, you know, because it's easy. Yeah. Got 3? Right, because you've taught them so well, Ronnie. They don't need so much help, because I'm not such a good teacher as you are. They need more help with biology. Yeah. So you can set yourself this plan at the beginning of the day, and then you can sit down, you can do 45 minutes. But there's a couple of rules when you're doing this. You've got to write down, and again, this will be in the web class, this will be a repeat. You've got to write down your task, single specific task. You can't just say, I'm going to revise biology for 45 minutes. No good. Okay, I am going to watch 4 videos and take notes and then do a past paper question on that topic. I am going to do all the suggest questions on the new spec past papers. I am going to try all the chi-squared questions from AQA and OCR, as opposed to doing my example, which may be at Excel. Okay, so like you're going to be give yourself a specific thing, 1 sentence to write down. And the other thing you've got to do is use a timer. And then put your phone away. Oh, you can't read that, it's terrible, but you can hear me. Use a timer. Yeah, you're going to then put your phone away. And when you've got focus, when you're actually doing one single task, you'll find that 45 minutes actually doesn't drag. The worst thing about revision is, A, it's never done, but this helps us plan, because once we've done these sessions, if you start this at a reasonable time in the morning, 45 minutes, 15 minutes, 45 minutes, 15 minutes, after you've done 2, maybe take a bit of a longer break. And then you do 2, 4, 6, 8, and then you're done. You might be done at 3 p.m., 4 p.m. possibly, and then you don't have to feel guilty that you're not working. It's a beautiful afternoon. The weather's gorgeous right now. Like, go and enjoy it, but only you can't enjoy it because you just get guilt, right? If you're just like, I'm going to work all day, and then it's like, oh, screw this, I want to go play football. Well, you're just going to feel shit when you're playing football because you know you haven't done the work. So set yourself these finite targets, do the tasks that you've set yourself to do, knock them off, and then when it gives you motivation to finish it, because when you've finished it, you're done for the day. Be like, peace out, I'm done. I'm over this. It's 4 p.m., but I've now got all evening to go and do whatever you want to go and do. Okay, sweet. What else should you do on the day of the exam? Again, I might go, this works for all examiners. My biochemistry lecturer told me this. My biochemistry told me this, and he said immediately before, 10 minutes before the exam, you want a small little piece of chocolate, like 4 squares of regular chocolate. You want maybe a Freddo or a Chomp. You probably don't want like a king-sized Mars bar, but you just like a small bit of chocolate, that little glucose hit is going to just peak your brain into like top performance. Do they still make Freddos? Freddos are like what I used to eat when I was at university. I just have a little Freddo on the way to the exam. Yeah, who's telling eat chocolate? It's great, but don't go too wild. And if it's like, if you're doing like a 6-hour art exam or something, then it's probably not going to help you because you're going to boost and then you're going to crash. But for a 90-minute exam, 10 minutes before, have some chocolate, make sure you're hydrated, all that kind of stuff. Also, just before your exam, this is genius. This is like, watch this TED Talk. Just search for Amy Cuddy. Her name is here, Amy Cuddy. It's your body language may shape who you are. This, you can see in the background of this, I think it's Usain Bolt, right? Tongue out, arms up, power posing, you know it. So, but there's studies on this. Blind runners, runners who are blind since birth, if they win a race, will do this with their arms. Well, they'll put their arms up like this. And okay, it's possible they've heard radio commentary of runners and it will say they've lifted their arms to the skies, but they're pretty sure that there's some inbuilt innate reaction to like putting our arms big and wide like this when we've done things we're proud of and we've achieved things. And so we can actually, not only do we do this when we experience those emotions, but we can actually reverse engineer it. We can do this and we can stand there and we can fake it till we make it. We can stand, she suggests doing this in the toilet cubicle. I wouldn't say, unless you can get your whole exam room doing it, go to the toilets, stand there, watch this TED Talk. She'll tell you way better than me, but stand there in a power pose, shoulders back, head up, arms out. And what actually happens to you during that 2 minutes is that your cortisol level drops, your stress hormone levels decrease, your ability to handle stress increases. You're then more composed. It slows your breathing rate down. Your parasympathetic nervous system kicks in. It means that people are statistically shown to improve their performance in interviews by just standing. When I used to teach languages, every 4 weeks we had a test and then they get graded and they go up a level, down a level. And I made all my students do this. And it's amazing. You don't go into that mad panic that you do when you're flustered in that first 5 minutes where you can throw away marks because you're not quite focused. This is going to really help you with that. I'd really, really recommend watching this video and trying it. Absolutely try it. What have you got to lose? 2 minutes in the toilet composing yourself before. Absolutely worth a shout. All right, questions. 10 minutes, questions. In fact, I might tell you to stop in a minute because I'm probably going to get swamped and I'd rather deal with a few, well, like I'll do like a minute per question. I'll do 10 questions, but I can't do like 100 questions in 10 minutes. It's not possible.
Rich?
Yep.
I've left some questions on Q&A that were like, that have been
biology related. I'll pop them open.
Yeah, biology or questions I didn't know the answer to.
I can go away. Spanish. You study Spanish too? I didn't ever study Spanish. I learned traveling in South America.
No, that's not a question that I left on there.
I've gone to the beginning, Ronnie. This is talking about Spanish.
What types of things do you recommend doing to get an A star in bio is the first one.
Okay, what types of things do I recommend doing to get an A star? If you're, no one has to know about outside the spec content. My actual advice, I'm going to turn my screen sharing off because you can just see me drinking some water. Stop sharing. Okay, so my general advice would be, go away screen share. Past papers, definitely. Using the past papers to figure out your weaknesses and then using the TT videos or your revision resources to target in on those bits. Work on the things you're not good at and target those. That's A star. You've got to know how it all fits together really well. If you're wanting to get A stars, I'd also highly recommend doing all the past papers from the other exam boards as well, because they're very similar. There will be things on there. If you're an A student, you don't do the ornithine cycle. But if you get a question that's like, this crazy biomechanical cycle that I've never heard of before, take it with a pinch of salt. Look at your specification. It's like, there's no mention of the ornithine cycle on my spec, so I'm just not going to answer this question. That's fine.
If I've already sent them, can you, does it look different to you? So the one at the top I've got now is I'm in year 12. If I buy the course this year, will I be able to use it next year?
Yeah, there's a few above that for me, but I can see that. So yeah, okay, I can see it. Yeah. Let me just do this and make them go away. Yeah. So if you're year 12 and you want to get the course and use it for next year, then you should send us an email, because we can set you up like that, but there's no option to do that on the website. So just send Travis an email, say I'm in year 12, I want to subscribe to the course, but I want to keep it for next year too, because the revision kits all expire on the 1st of July.
Is there a practical biology video?
100%. There's a full course on all the practicals. In fact, I can share screens and I'll show you. Oh, it's over here. I think I had it in Firefox. Okay, so this is a test account that I have for me. This has the biology revision kit. So if you buy the biology revision kit, this is what your My Courses page will look like, and you'll see the practical skills course here. And then it's divided by exam board within this. So you've got all the required practicals for AQA, 1, 2, 3, 4, 5, 6, 7, 8, however many there are. And then the OCR ones using a light microscope, the photometer, chromatography, RF values, gel electrophoresis, EdXL practicals. There's more for EdXL. There's 18 required practicals for EdXL. And how this is one core practical 9 versus core practical 15, almost identical. Tangents and how to calculate the rate. So the full course is covered. All the practicals are in there in the practical skills course.
Okay.
Scientific article. I'm doing a full web class on the scientific article. So if you're interested in EdXL doing the scientific article, then basically get the revision web class series, and I'll go through that with the paid web classes.
Bullet points. Couple of questions about using bullet points.
Okay, so you can use bullet points in every answer except those ones that are telling you that you're going to get marked by level. If it's an asterisk in OCR, AQA, you can use bullet points for everything except the essay. And again, if it's marking you on your level answers, then you can't write in bullet points. But if you are writing in if it's general, describe or explain 3 or 4 marker, then you are welcome to do that. You're welcome to write bullet points. Synoptic essay, again, I am doing a full web class on the essay. So buy the revision web class series for biology, and I will go spend a full web class going through all the planning, how to get more marks. My AQA essay plan came from Ray Ski, who's was chief examiner for AQA A-level biology. Do we get the crash course as part of our membership? Yeah, so if you buy the revision kit, you can access all of the biology stuff, basically, and you can go through, as I said, people have completed the course. The fastest was 2 days, but I wouldn't recommend smashing it in 2 days. Okay, I'm at the top of the list now, I think. Every time a question gets added, it moves off my screen. Okay. Is this web class... Are the old spec papers? Yes, I definitely use the old spec papers. Question there. I do all the new spec papers first, do all the new spec papers at least twice, but the actual content of the old spec papers is nearly the same. You will find that you'll effectively get just as much value out of them. The style has changed, but the content for the vast majority hasn't changed. If you struggle with a particular topic, go back and use the past paper library in the course, download all the old spec past papers, and again, go through them and Command F, Control F, search for those key terms and those questions you know are going to be tricky on that topic, and hit those ones. Am I doing A2 content? A2 content is the web class tomorrow, free web class tomorrow. How do I get pedantic questions when the answer seems too simple or obvious to answer? Oh, it keeps going off my screen. AQA have a couple of past questions that are really annoying when it comes to the mark scheme. They're all basically really annoying and pedantic when it comes to the mark scheme. So in your past papers, analyze your past paper questions in the same way I've shown you how to do tonight. So analyze those questions and then mark the question immediately afterwards. And what you want to be trying to do is learning the mindset of the examiner. That's what you really want to be trying to do. You want to do the question, mark it. You want to then add in all the bits you didn't get. And you want to do a bit of analysis. It's like, okay, what did this question really mean? So spending the time in your past papers to learn how they answer the questions is most valuable. Are you marked down instead of bullet points in continuous prose? No, unless it's a continuous, unless it tells you that you need to write in continuous prose, you can write in bullet points. There's a full web class on the essay. I've covered that one already. ultimate. So anyone with the paid course gets, I showed you, I think it goes over here. So anyone with a paid course gets the past paper library, and you go in here and you can basically see, you can more or less download all of these folders are the old spec, the new spec, all the past papers are in there. Are you going to get an A star? If you complete my course, then you're highly likely to. I can't answer that. I don't know where you're at. Do you need to know about the Apoplast and Simplast pathways? If you're asking me questions like that, you're going to have to give me your exam board, because no, if you're an AQA student, because they've just removed that from the spec. Are there any new specimen papers for biology or chemistry this year? There's not new specimen papers. They only release specimen papers just before the spec is released. They're the exemplar papers. So there's no new specimen papers, but you've obviously got last year's paper, the 2017 papers. How do you know? I'll show you. I think I've got an example. Are you still seeing my screen? I've like moved off. No, okay, let me share. Let me just figure out what's going on here. Desktop 1. Why is it not tracking me? Okay, I'll just drag it across. I'll go and grab it and drag it across. Okay, you should now be able to see that, right? Yeah, you can see my window here. So the question is, how do you know? New spec OCR. What exam board do you do? If you're AQA, you don't have to worry about it. Let's go. I need a question paper. Yep. There should be one in here. Won't be in the multiple choice section, that's for sure. Oh, did I just see one? In fact, it's probably going to be a 6 marker. I'm just going to search for 6. I've got so much stuff on my screen. Square bracket 6 then. Telling me there's no 6 mark questions on this exam paper. I don't believe you. So all of these questions you can use bullet points. All of these ones you can. It will tell you additional space. There's an asterisk that denotes the question. That's the chat. I'll find it. Okay, let's go. Let's nitrate ions looks like a nice answer. Why is the xylem not affected? I'm assuming that was the metabolic inhibitor question, because xylem is dead. It doesn't matter if there's ATP or not ATP. It's transpiration is evaporation through the stomata. And basically you've got water vapor leaving through the leaf, which basically creates low pressure from the top. And then you've got a straw, which is the xylem, basically this hollow tube, and it's just going to suck water up, literally the same as sucking on the top of a straw. That's why the xylem doesn't need ATP to suck water up. But it's also the reason that it can only go one direction. It can't push stuff down because that requires energy in the form of ATP. Serial dilutions, again, serial dilutions, Zara, fully covered in the course. Every part of the course is covered in the course. So if you want the serial dilutions bit, then that will, we're in the past paper library. That's all. I've just been logged out. Interesting. Okay. Can you lose marks for answering with too much information? No, you will never get marked out with too much information. You will waste time though, potentially. If you're going in, if it's an outline question and you're going into all the little details, then you're just going to kill yourself off by taking too much time. You don't do A-level maths and struggle with the maths. What's the best way to answer the maths questions? Again, there's a full course on the maths and stats element of the course. I can't go through all of it now, but that goes through count all the things, the percentage change. The most common maths questions you're going to deal with, percentage change, calculating the rate. That could be breathing rate, could be heart rate, it'll be a cycle, and it will say you have to work out probably from a graph that one cycle takes 0.85 seconds, and therefore you have to do 60 divided by 0.85 to give how many cycles there are per minute. They're really common. Surface area of a circle and volume of a sphere.
4/3 pi r cubed.
4/3 pi r cubed. See, Ronnie's on it. They're the ones that you could be asked to work out the surface area of a cylinder. Yeah, I mean, the only way I could assume that happening if they give you like a rod-shaped bacteria maybe, but then it would have rounded ends, and they'd have to make some pretty major assumptions about the bacteria itself as to, because you can't calculate. Well, you could do 2 hemispheres and add it on, but that's not a question that they're not going to go into that much detail in A-level biology. It'll be probably volume of a sphere, surface area of a circle, percentage change, or the rate change, possibly some log scale. I'm not gonna. Am I single? Was that you, Ronnie?
Oh, no, that was to me. Thank you.
Oh, was it? Yeah. Oh, who was that to? Who were you asking, me or Ronnie? How can you be sure your points are relevant? Okay, so understanding what the topic of the question is by analyzing the question, especially in your past papers, it will slow you down initially to interesting. I'm on. What's going on? I might just mute that for a second. You can still hear me? I assume you can, Ronnie, if you can still hear me.
Yeah.
It's happening everywhere. Just someone's trying to get hold of me that badly. Everything's on Do Not Disturb. I don't know why it's coming through. Okay, so yeah, how can you make sure your points are relevant? You can basically make sure that you are using the past papers to analyze the question and then brainstorming the key terms on that topic. And you're then going to pick the most relevant 3 to 4 key terms, and then you're going to put them into your answers. Paper 3 questions is a full web class on paper 3, so we're going to cover that in the paper 3 web class. Grade boundaries, you don't need to worry about grade boundaries. All your task is get as many marks as possible. There's no point stressing about what the grade boundaries are going to be, because it's purely going to be how many marks do you get. If you get more marks, you're going to get a better grade. It's really that simple. There's nothing you can do about the grade boundaries. All that you can do is prepare yourself as best you can by doing the past papers, using the videos, and then smashing it in the exam and getting them more marks than you possibly could have done any other way. AQA is very similar to WJEC. Yeah, absolutely. One of my good mates from uni now teaches WJEC biology, and he's actually been through my course and was like, there's a few, you do chitin in WJEC and you don't do chitin in AQA, but more or less it's the same. How many maths papers should you do a week? Are you talking about the maths in biology? If we're talking about maths in A-level maths, then you can save that for Ronnie's web class tomorrow.
Yeah, she's doing 6 papers.
What's the best way to remember the content? Diagrams, summaries, active recall, testing yourself, spaced repetition. They are the best ways of basically remembering anything. But by testing yourself and then leaving a gap and then testing yourself again, that active recall with what's called spaced repetition is the best way of building it up. Do diagrams, do summary diagrams, do flow diagrams, draw it by hand, because if you're just working, like there's lots of research on computer-based learning versus paper-based learning. Typically people who are writing on paper do better in terms of recall, not because there's something that when you're working on a computer, you can type typically faster than you can write by hand. So when you're writing by hand, you've got to do more synthesis in your brain. You've got to prioritize which bits am I going to write, which bits aren't I going to write. And if that process, that mental rejigging of the information in your head is what's building synapses. The fact that if you can touch type at the same pace as someone's writing, then you'll hear them and you'll just type it out, but you're literally a machine at this point, just regurgitating numbers or letters. You're not processing any of that. If you're taking notes from someone who's speaking verbally, you can't write that fast. So your brain has to work as best it can to pull, okay, what's important? This, that, the boom, boom. And that work, that mental energy you're investing is the learning taking place. And that's why writing things by hand and creating by hand is more effective. Okay, last 2 questions. The practical content, get the course. It's all in there. That doesn't count as a question. If you have an afternoon exam, I probably would do some revision in the morning of an afternoon exam, and it'd be kind of crazy not to, especially if there's a one top problem area where you could like add a little bit of extra knowledge, then that would be one thing to focus on. Recapping your key terms on those topics, because remember the key terms are basically the answers in terms of how to get the how to get the marks in the exam. I'm going over the essay in the web class, so check that out. That's what I do in the morning of the exam. Are the equations given? Yes, except you're given all the stats, all the t-test, the chi-squared, correlation coefficient, standard deviation, although OCR did cock up last year and didn't give the equation sheet, but that was a mistake from them. It wasn't, they didn't deliberately not give it to you. You do need to know the volume of a sphere and surface area of a circle, pi r squared, 4/3 pi r cubed, all that kind of stuff that you do need to know. So you could spend a session just learning some of those formulae.
Rich?
Okay, I think we'll do one more, and then I'll just give you a little reminder. Don't forget all those competitions. I'd love it. Like, I'd love some student testimonials. In fact, do you want to do it now, Tom? In fact, you can tell me that in the chat if you're down for reviewing. Again, I have no idea what you're going to say.
Have you told them the code?
Okay, Tom, I'll find you in the list. Do you have a microphone and stuff plugged in? Oh, you don't need a camera. I won't turn your camera on. You just need a microphone. You do. Right, let me find you in. Where are the participants? Over here. Tom. Okay. Now you're going to be over here on the panelist side. Okay, I'm going to turn your microphone on. Can you hear me, Tom?
Yep. Can you hear me?
Oh, I can't hear you. Oh, I've got the microphone coming up and down.
Hello?
I can.
Although
I can
we
hear
don't
Tom.
hear anything.
Yeah.
Ronnie, you getting anything?
Yeah. We can all hear him.
Although, oh, hang on a minute. It's because my Mac's muted. That's why, because I was ringing. And can you hear? Can you say something else, Tom? Because I probably can hear you.
Hello?
Hey, wonderful. Yeah, it was my incompetency. I just muted it when my phone started ringing. Okay, so Tom, what have been your experiences of using the Taylor Tutor's biology course?
Basically, I've used it for the last year. It's been the best money I've spent, and all the videos, they basically cover the content that you'd be reading a textbook for a couple of hours, or you could just watch a couple of your videos and that'd be 20 minutes and you'd learn exactly the same stuff. And it's so concise, and it's the best way to prepare really for the exams. So no, it's been great use of my money and great use of my time, to be honest.
Awesome. Do you feel like the way that the videos give you the information, do you feel like that helps you with your exam answers? I mean, I know that A biology is super tricky because of those specificity of how you've got to use the words on the exam. Do you find that the videos help with that?
Yeah, I mean, it's like going through all the videos, it's really clear that you've gone through all the mark schemes, because if I go and do a paper once I've watched one of your videos, I can literally see the answer in the paper on your video. Like, you don't cover any of the waffly crap that like the textbooks go over. You literally write the stuff that is going to be in the mark scheme. So it kind of makes me think more like the examiner and what they want, rather than the stuff that's in textbooks.
Yeah, I mean, like, it's not a coincidence, obviously, that I trawled all the past papers. I did a lot of work, and I use the words that are going to be in the exam. If the question comes up on, I don't know, DNA replication, then that's exactly the words that I've chosen to be in there.
Yeah.
I can't see, Ronnie.
1/3.
Oh yeah, you're quite right. Okay, yeah. So what I've totally forgotten to tell you all.
9:40.
And I probably should have done this at the beginning, but you're all going to come back on Saturday anyway. So thanks, Tom. I think that's awesome. I was told, did I set you up for that in any way other than just asking whether you'd be willing? Like, we didn't speak before this. I didn't mole you in here.
No, it's all genuine. All genuine.
Cool, man. I will turn your microphone off. Thank you very much. I'll see you tomorrow in all the A2 answers in an hour. I'm going to go through very similar to the one I did on Tuesday. I'm going to rip through as much of that content as I can. And yeah, we will, I'll be in London tomorrow, so I don't quite know where I'm going to be. I'm going to be in Old Street visiting. Tom, are you coming to that session in London? You've been...
Unfortunately, no. No, I live in Kent.
Where about? Oh, okay, you're a bit far away.
Yeah,
well, I'm sure at some stage we'll get a chance to check in. Right. Okay, so thanks, Dan. You are muted. Okay, so the last thing that I am going to leave you with is basically, if you are thinking that you would benefit from using the course over the next month before your exams, we can give you basically a bit of a deal. I can turn Tom off from being a presenter though. How do I do that? Panelists is here. Oh, I've got so much going on. I can... Boom. So, okay, so until Sunday at midnight, we're going to offer you a deal. We're going to offer you a deal. And are you seeing my screen? No, you're not seeing my screen. Okay, so those of you who's interested in getting the course, I want to just see it in the chat. Who would ideally like to have it? Wonderful. Okay. Why? Who's gonna buy it? Who's like decided they're gonna just buy it anyway? It's not, if it's cheap. Okay, well, value for money, I have zero. I can look you dead straight in the eye. I can tell you that there's absolutely every penny's worth of value in there at full price. But we are going to offer you a bit of a sweetener. Who's doesn't think they're going to buy it? Going from an E to a P is definitely worth it. Okay, so this is the deal. This is the deal. The deal is you go and speak to your aunts and uncles and grands and grandfathers and your mother and father and your brothers and sisters if they're old and grown up and got loads of money. But basically, if you are prepared to cover 1/3 of the cost of the course. So the biology course covers the entire spec, which is why it's a little bit more expensive than the maths and chemistry revision kit. The maths and chemistry revision kits are revision courses, which are going to cover the vast majority of it and are going to super help you out with your grade, but they don't cover the entire specification this year. They will next year. So the biology course is £120 full price, of which it will transform your grade. But if you can cover 1/3 of that cost, then your parents or your family or your friends or your relatives cover the other 1/3 of the course. And we will cover the final 1/3. So if you can find 1/3 of that, £40, I would suggest what you go and you say to mum and dad, like, okay, I'll do the washing up, I'll wash the car, I'll mow the grass, I'll hoover the floors. Like, if you genuinely show them how much value it's going to give them, and again, share some of the stats on it. Students who completed it last year or completed, I think, more than half of the course. I think it was like, maybe it was 70% of the course. If you complete 70% of the course, 50% of students went up 2 grades or more. 17% of students went up 3 grades or more. It works. I know it works because I've seen it. Hundreds and hundreds of students. Like, look at those Facebook reviews. Like, they're not made up. Look at those reviews. Like, read them. Go and show your parents the Facebook page and be like, do you think it's worth trying? So £40 from us, £40 from you, and £40 from your folks. Like, if you wanted me as a tutor, you wouldn't get me for 1 hour. When I finished tutoring, when I finished my one-to-one tutoring career, I was £200 an hour. And so you wouldn't even get 1 hour of me for the price of the full course. And right now you're going to get every single one of those lessons. Took me about 2 hours to plan, to plan. And then I had to sit down, film it, had to get it edited. I don't know how many hundreds of hours of my life have gone into building that course. It's been a huge amount of work. And I know it's successful, and all I really need to do to make it, I can't give it away for free because I've spent my entire life, for the last 3 years of my life doing this, I have full-time, wonderful, wonderful teachers. Like Ronnie's assistant principal examiner. What's your official title? Assistant principal. Ronnie writes the
examiner for the new A-level paper 1 and paper 2 and the old A-level C3 and C3-4.
So like she writes the answers for them for Excel maths. Like she's like, yeah, she's a top quality, wonderful professional, but she doesn't work for free. She loves me, but she doesn't work for love alone. Yeah, I mean, I would say go and have that. All that I ask you to do is go and have the conversation. Go and have the conversation with your mum, your dad, your grandmother. And it's not like you're asking for a new pair of jeans or a new computer game or something that you are just going to enjoy but like not get any value from. This is going to effectively change your grades. It's going to get you into that university. Do you think is it worth getting to university for 40 quid, a few extra hours working? There's so like the biology course covers the entire spec, all the practicals, all the maths, all the stats, all of topics 1, 2, 3, 4, 5, 6, 7, 8. It's all in there. Like I will basically be able to sit down next to you and teach you any single topic that you want help with. Any single topic.
Hey, Rich.
Yo.
You didn't tell them what the code is.
Oh yeah, I can tell you what the code is right now. So yeah, I'm super distracted, but basically come on Saturday and I'll give you a way better description. The coupon code is 1/3, and I'm actually just going to show you basically 1-3 is what you need to stick in. I'm going to screen share with you and I'm going to show you what's going on. Okay, let's do this.
It's not only one course, it's all the courses.
Yeah, and this would actually, you can get this off the biology course, off the maths course, off the chemistry course. Rich did a way better job of this than I did. I'm making a right mess of it. If you go to, in fact, I'm going to do this in Firefox, because if you go to academy.tailoredtutors.co.uk, this also applies to the All Access Pass.
They're talking about all 3. You can get all 3 there.
Okay, so if you want to buy more, if you want to buy just 2 subjects, it's just you can get 1/3 off the maths and 1/3 off the biology, and you can get that. But if you want all of the course, full access to everything, all the biology, all the maths, all the chemistry, then the All Access Pass is where you need. It's basically going to be the full price, with including all the tax and whatnot, is 300 quid. So we're going to give you £100 off it. So what you have to do is go to add coupon, and it's 1-3, hit apply. Oh, because I can't spell third. And it's £100 off. If you get this course, you get the biology, the maths, and the stats, but we're also going to throw in all of the web classes. So you can join all the paid for, the small group maths web classes. So you'll have like a small group with just Ronnie for an hour and a half to answer all your questions. You'll get a small group with Richard doing all the chemistry stuff. There's not going to be 300 people in these rooms asking questions left, right, and center. It's going to be a small group. Basically, every question gets answered during those web classes. You get all of AQA paper 3.
You don't get access to the web classes if you're just buying them individually. If you just buy the maths or just buy, that's only.
Yeah, if you just buy the biology revision kit, you just get the biology revision kit. The only way, I mean, this is basically 200 quid for everything. You get everything we're doing for that price. So yeah, it's including tax. It's going to come in at £200 for everything. It will be the biology, the maths, and the chemistry, all the web classes, everything. Am I a millionaire? Do you know how much money I spent building this course this year? Definitely not a millionaire. You know how much I've paid myself building this course? How much per hour? I've been well under minimum wage.
And he hasn't paid me anywhere near enough.
No, definitely not. Any questions for us? Any questions? You can try it out this weekend. You'll see this weekend. If you invite some friends with the All Access Pass, you can give it a go. It will just give you a taste of how useful it's going to be. Is the 200 valid for 2 years? No, it's just for this year. Do I do private tutoring? You want to pay me £200 an hour? For 200 quid, I'd highly recommend you buy the all access pass. You're going to get way better value out of that than you're going to get from, there's only so much I can do in 1 hour, as good as I am. Yeah, the same coupon for all 3 subjects, 1-3rd. Essay questions, I'm going to do in the essay web class. So if you join the web classes, then you will get all my top tips on the essay questions and any predictions. So if you're an AS student and you want it for next year, send us an email. There are options for that, but it'll be totally different basically. So send us an email, say you're an AS student, you'd like to start, you'd like the videos now, and you'd like them for next year. You'll get a fair bit of extra stuff for free, basically, by doing that.
The coupon is until midnight Sunday.
Can you buy 0.02% of my business? How about no? How the coupon is valid until Sunday night at midnight. It will, Monday morning, she's gone. Advice on keeping hydrated but not needing a piss during the exam. That's a good question, actually. Okay, so practice it with past papers. Practice, so you know your timing of the past paper. Plan how much water you're going to drink beforehand. If it's going to be a 1 p.m. exam, go do a past paper at 1 o'clock under time conditions and see for yourself. I don't know how big your bladder is, but you can set yourself up, do a 9 o'clock exam, get up, drink a pint of water, and then try the test. Is that too much? Is that not enough? That would be my build a strategy for you. Can you use the coupon on the web classes? No, it's just off the revision kits. So, I mean, it's insane value as it is. If you want to get all the kit contents and all the web classes, you've got to basically buy all 3, or you can buy the revision kit for £1/3 off and then add the web classes for £30. 30 quid is like a standard tutor, like not a particularly good. A standard tutor is going to be £30 an hour. So effectively for 3 hours or 4 hours of tutoring, in fact, we're going to give you £1/3 for a couple of hours of tutoring with a standard tutor, you're going to get the entire course taught to you by me. And
why did you buy Snap Revise? You're all saying I shouldn't have bought it. Why did you buy it? Because I looked at their advert and I wasn't 100% convinced.
How much did you spend on Snap Revise? How much was Snap Revise? £50, 25 quid, 25 for one course.
Thanks.
Ronnie is amazing. Yeah, way better. Is it Charlie? Who's the maths Snap Revise?
Jack.
Well, tell all your buddies.
The maths course that I've made is C3, C4, but I'm doing 2 hour-and-a-half webinars on, thank you, David, on M1. I think the first one is next week, actually. I'm doing M1, S1, S2, loads of C3, C4, but also M1, S1, and S2. Oh, and D1, I've got some videos. If anyone wants the D1, then I will give you a link to the videos.
Doesn't feel like you're actually learning.
Can you email me, maths@tailoredtutors.co.uk, if you want the D1, okay? And I'll send you a link.
Okay, so what's the coupon? I'm asking you what the coupon is. 1-3rd. Doesn't matter if it's capitals, lowercase, doesn't matter. What courses is it valid on?
Hey, Rich, can we have that quote? I've learned more this week with Tailored Tutors than in a year with Snap Revise.
Yeah, you can put that as a Facebook review. That would be wonderful. Yeah, you're welcome to. Again, you can win the course for free. There's right now, or as of this evening, I think there were 6 people who've written a Facebook review. That gives you a 1 in 7 chance of winning it for free. We'll just give you. I tell you what. for the Facebook review, the winner will give the all access pass. They can have everything. So if you're super into it, you've probably got like by the time the end of the week comes, maybe 30 people will have done it. So like leave a Facebook review, make sure it's public. Again, we'll pick you at random. We're not incentivizing you to leave a nice review. I want you to leave an honest, like people are going to respond to an honest review way better than just a phony review. So like write a genuine, honest review. We're going to pick someone at random and you can have the winner of that one can have the all access pass. So make sure you attend the web classes on Saturday because we'll give you all the inside information. There's going to be like that web class I've titled How to Improve 2 Grades Without Doing More Work. So like how to use the past papers, how to spend your time focusing. And then this is a month to go. This is go time. And like I work in this is, well, it's all of our fields, right? So, but like I work a lot in memory, learning, productivity. Like this is one of the things that between us, we can give you loads of top tips, which is going to change the way that you use that month. In a month, you can add 2 grades in a month if you're using your time wisely.
Definitely.
2 grades, definitely.
Who's coming to do integration tomorrow with me? Yes, sir. Excellent.
And go and, all I'm asking, go and have a chat to your folks, to your parents, to your guardians. Go and phone your grandparents and go and mow their lawn for them. Like if they can see how clearly it's going to be useful to you, like it really is. It can't be made any cheaper. That you will not get better help for better money anywhere online. That I can tell you for sure. So I can show you, in fact, what I can show you is just like the Tailored Tutors website. I can screen share and show you that exactly what it looks like and what you get. You can now sort of dig around and have a look. Oh. Luckily, it was only AQA's email address on the screen.
Lucy, the web classes, the list of when they all are is on what Rich is clicking on now.
Okay, so like if you want to, so these are the kits basically. The biology kit, what's included. If you want to have a look around and poke around inside, you can do the view kit contents, and then you can see all the courses that are involved. You can have a look into the maths and stats course if you want to have a look in there, see what's included, yada yada yada, and have a look. So that's where you can sort of poke around and see what you're going to get. It's where you can show your folks and all that kind of stuff. If you want to look at the Maths Revision web class series, you can look at the Maths Revision web class series. It'll give you what's all the dates, all the times, what's going to be when, all that kind of stuff. So that's where you find all of that. Again, if on the packages and pricing page. So the 1/3 promo is off the biology kit, the chemistry kit, or the maths kit. The web classes are not included in that. So if you want to add those as extras, they're £30. Unless you get the all access pass, in which case you get absolutely everything on this page for £200, which used to be an hour with me. And now that was basically, yeah, that was the deal. Okay, I think we're more or less done. I've lost the chat. So stop sharing, come over there. I think my chat must be in another window. Thanks, guys. You guys are awesome. We will see you. I'm both. It's Ronnie and I tomorrow. Ronnie at 6 o'clock. Yeah.
Tomorrow, you're 6, I'm 8.
I think it's the other way around. No.
But oh yeah, no, it's the other way around. I'm 6, you're 8. Yeah. 6. Yeah. I'm
in London with the doing some feedback with the pay students tomorrow.
I'm doing 6, which means you're going to kick me out after an hour and a half.
Yeah, an hour and 45. I'm going to be touch and go ready for that session. So yeah, you can do an hour and 45 if you like.
Thanks. Yay.
Awesome. Okay, thanks guys. See you all tomorrow, or yeah, you should all be here tomorrow because I'm going to teach you some more awesome stuff. Bye.